All right, so next up we have Dr. Sean Adams and a hypothesis that won't go away. Does lipotoxicity contribute to myopathy and FAODs? So joining us for the first time on our agenda is Dr. Sean Adams. Dr. Adams is professor and vice chair for basic research in the UC Davis Department of Surgery. He also serves as the founding scientific director of the Center for Alimentary and Metabolic Science, a research group established to promote cutting-edge science that is relevant to human health and clinical care. Prior to joining UC Davis, Dr. Adams was professor and chief of the developmental nutrition section in the Department of Pediatrics at the University of Arkansas for Medical Sciences. He served as director of the Arkansas Children's Nutrition Center, one of 6 national USDA-funded human nutrition research centers, and has a mission to understand how maternal child nutrition and physical activity optimize health and development. Formerly, Dr. Adams was the research leader for the Obesity and Metabolism Research Unit at the USDA ARS Western Human Nutrition Research Center, an adjunct associate professor in nutrition at UC Davis, and before that, a researcher at pharmaceutical companies Genentech, Novartis, and Amylin. In leadership positions since 2008, he has overseen research programs totaling approximately $60 million. His personal reach is focused on identifying the molecular underpinnings of metabolic health. His lab also studies muscle lipotoxicity, metabolite-associated inflammation, exercise physiology, and the role of myoglobin in fat metabolism. Dr. Adams has authored over 120 peer-reviewed scientific papers, is associate editor for Advances in Nutrition, and a member of the National Institute of Health, Nutrition, and Metabolism in Health and Diseases Study Section. He has given over 80 invited talks in the U.S. and abroad. His academic training includes degrees from Cal State University, Fresno, UC Santa Cruz, and the University of Illinois, followed by postdoctoral fellowships in Barcelona, Spain, and UT Southwestern Medical Center in Dallas. When he is not doing research, he and his wife Sharon like to spend as much time as possible exploring the great outdoors and traveling. Their son Connor is a graduate student at UC Santa Cruz, and his brother Nathan is a member of the Colorado Air National Guard. Please join me in welcoming Dr. Adams.
Well, thank you very much, Kyra. That was, now everybody knows all there is to know about myself and my family. So that's I don't know if that's a good thing or not, but thank you all. I also wanted to thank the organizers for this opportunity, especially Stephanie Harry for reaching out to me originally and having some conversations. This is not my main area of research by any stretch. I've been the beneficiary of learning from everyone today and in previous meetings where I've had an opportunity to interact with families and researchers such as Melanie and Jerry. I also want to thank, leading into my talk, Dr. Gillingham, Dr. Corson. They've given a really, I think, beautiful overview of bioenergetics and metabolism. And so some of the crazy graphs and figures that I'll show you today will make immediate sense to you because you've already benefited from their talks already. So I just want to check that everybody can see the screen okay and can see the laser pointer. Does that look okay, Kyra? Okay. So why do I say a hypothesis that won't go away? Well, again, this isn't, FAODs are not something that I ever thought I'd be working on, but when we were looking into type 2 diabetes and the role of muscle and mitochondria in sort of diabetes, cell stress in muscle and heart and these sorts of tissues, we couldn't get away from the fact that what we were learning might have some applicability to FAODs as well. And so while I'm not going to convince you, I don't think today, that we have the answer to rhabdomyolysis or myopathy, cardiomyopathy, and general, sort of malaise that occur episodically in people who are affected with these diseases. I don't have all the answers. I'm hopeful that we'll at least open some new doors of thinking about what's the actual molecular basis for these episodes? What might be contributing to them? And if we could figure that out, I would reason that new drugs or new approaches, whether they be nutritional or pharmacologic, could be implemented to intervene in those specific spots in the pathways. So we've been thinking about this a long time, and it just won't seem to go away. We still keep coming up with new data. So with that, if my computer decides to agree with me, there we go. So the question of the day is really this: do specific metabolites that accumulate in FAOD trigger pro-inflammatory and cell stress events? I would argue, and I'll say this at the end of the talk, I don't think it's any one thing. And you've probably been sort of capturing that today when you've heard the other speakers speak. There's probably a set of triggers or a very specific context that must take place that causes a cascade of events that lead to somebody ending up in the ER, for example. We heard the stories from Tasia earlier. So one question I think is very reasonable would be, are there metabolites that specifically impinge on different biological pathways that might be contributing to this problem? And the way we've looked at this question in diabetes and then leading into FAODs is very, very simplified. Here's a mitochondria where fuels are coming in, oxygen is consumed, and you heard about ATP earlier. ATP being the currency of energy, which supports cellular work, functions, and metabolic pathways. So when all of this is working, sort of perfectly, ATP is made, work is being done, the cell is happy, the cell is healthy. We've been questioning, what about the exhaust? No system is perfectly efficient. And so as this metabolic machine is operating, are there exhaust products, if you will, that could be playing a role in impacting the health of the cell and maybe causing inflammation or causing cell stress when they accumulate in excess? This is our simplified model. I've been showing this slide for many years. It hasn't changed, and I don't think we have all the answers just yet, but I'll show you some examples today and try to convince you that we may have stumbled upon, at least a set of metabolites that could be part of the exhaust and are important in these diseases. The way I look at it, again, some of you may have seen this slide before at the Inform meetings and such, but seems as if, so we can think of efficiency of fuel combustion in that mitochondria I mentioned to you earlier. Here's a car that gets supposedly 261 miles per gallon, and so you can imagine it can go a mile and have not a lot of exhaust. And you can compare that then to my old car sitting in the garage, and there's my son. This is the National Guard guy you heard earlier, by the way. This one doesn't get particularly good gas mileage, I assure you. And so you can go the same distance in these two cars, but certainly this car over here is going to have a lot more exhaust compared to this car. And the question would be, does this happen in us as well? And are there contexts in which the exhaust is particularly prevalent and can cause issues metabolically? So you've seen some of these slides before. I wanted to start us out with a primer just to remind us where some of these metabolites come from. And keeping this as simple as possible, in the cell we have the non-mitochondrial space, or the space outside the mitochondrion, shown here. And then here you have the space within the mitochondrion, shown here. And you've heard about some enzymes earlier: carnitine palmitoyltransferase 1, carnitine palmitoyltransferase 2, and this sort of carnitine acylcarnitine transporter here. When fatty acids come into the cell, they're converted to what's called a CoA ester. CPT1 can convert this then to a carnitine ester, and we were hearing more about these acylcarnitine esters earlier as biomarkers of metabolism and diagnostic biomarkers. Long-chain fatty acylcarnitines can then enter the mitochondrion, where they're retroconverted back to their CoA ester, undergo beta oxidation through several rounds that I think Dr. Corson described very, very well, where these long chains are shortened bit by bit to end up with a 2-carbon acetyl-CoA here at the end. They that can enter what's called the Krebs cycle or the TCA cycle, where it further gets combusted. Along the way, there are these metabolites here that enter what's called the electron transport chain in the mitochondria. And then this is what is the fuel, which is fueling the ATP generation that Dr. Gillingham mentioned. So when this machine is burning efficiently, this whole process moves forward very efficiently with very little exhaust. In the end, certain metabolites can be converted, as I'll show you in a minute, to different carnitines and different fatty acid esters. And of course, when you have a fatty acid oxidation disorder, it can occur sort of toward the end of the process, in the middle of the process, or at the beginning of the process, and this will influence which metabolites are generated at any one time. So here's just a general view of an acylcarnitine. The carnitines shown here red, the acyl group or the fat group is shown here in green. We can think of acylcarnitines as being sort of surrogate markers for metabolite accumulation. For example, if we start with a long-chain fatty acid here and we go through fatty acid beta oxidation, and remember, as you all know, fatty acid oxidation disorders can occur in many of these places here and can back up the system. At many parts of the system, acylcarnitines are created or converted from their CoA ester from these pools of metabolites that are in the cells. So for purposes of discussion, we want to think about muscle and we want to think about heart. However, I would remind us that these sorts of processes go on throughout the body, including the immune system, and that'll become important later as I start to kind of throw some hypotheses your way that maybe the immune system is also involved in some of these pathologies that we see that can transpire episodically. Carnitine acyltransferases are the enzymes that convert these metabolites to these metabolites. And so I guess the point being, acylcarnitines generally report on the status of metabolism at any one time. And that's why they're so important as biomarkers or diagnostic tools. The question will be a little later, are they also having bioactivities of their own? I mentioned earlier the Krebs cycle or the tricarboxylic acid cycle. Some of you may be familiar. And this is an oversimplified version. But as fatty acids are burned and produce those 2-carbon metabolites, they enter what's called the Krebs cycle or the tricarboxylic acid cycle. And all of these things, when operating efficiently, can generate reducing equivalents and CO2, which we breathe out, as you know. And along the way, ATP is produced. So between beta oxidation as well as the Krebs cycle, you can combust fuel generally pretty efficiently, and then this can help to produce ATP and support normal function in the cell. However, something that's very interesting, whether you are afflicted with an FAOD or not, fat combustion is actually pretty inefficient sometimes, and this is very natural. And I'll show you some examples of that, and we'll start to peel back the onion, so to speak, to understand why that might be the case. So if we track radiolabeled palmitic acid, so this is a long-chain fatty acid measured with a radio label that we can track very, very carefully, and we take muscle mitochondria, we can look at the fate or where this radio label ends up, whether it's in CO2, which would be reflective of complete combustion in the Krebs cycle, very efficient, like the Volkswagen I mentioned to you earlier, or whether it ends up in these sort of byproducts, which we call acid-soluble products or incomplete beta oxidation products. That would be like my T-bird I showed you earlier. And what you see is as you increase the concentration of fatty acid here, it actually turns out you're increasing fatty acid combustion in these mitochondria as well. I don't show that here. But what's interesting is, in lockstep with increased fatty acid combustion, you also see an increase in these sort of incomplete beta oxidation products, reflective of incomplete or inefficient fat combustion. And these are from mice, just normal healthy mice. Here are humans during exercise. If you track the acylcarnitines in blood, These are the plasma concentrations here as a percentage of time zero. This period here is during an exercise bout at about 45% of maximum. So you may recall Dr. Gillingham's talk. She mentioned this is the sweet spot for fat burning during this time, and then a resting period here. And the red and the black are just two different conditions. It's not really relevant for this particular point. But these are medium chain and short chain fatty acylcarnitines in the blood. And so what this means is these are sort of the exhaust or incomplete beta oxidation, where the long chain fatty acid has been chopped down to smaller bits and then is not fully combusted to CO2. It's reflected in the blood very clearly that exercise causes a massive increase in these chain shortened, inefficient sort of markers of fatty acid oxidation. So the point being, this is very natural. We have acylcarnitines are always going to go up in the blood depending upon the exercise level of the person at any one given time. These then drop pretty precipitously with rest. Okay, so the take-home message really is that when there's a mismatch between the fat availability or delivery to the tissues versus combustion, this can lead to sort of smog of lipid derivatives, including acylcarnitines. So we know this, of course, for FAOD in certain situations in which acylcarnitines can rise very high in tissues and in blood. This is a reflection of a sort of a mismatch between the fatty acids within the tissue, delivered to the tissue, and the ability to combust those fatty acids. So what contributes to inefficient and incomplete fat oxidation in, say, a non-FAOD situation? Just sort of say in me, for example, when I start to exercise, why would I have incomplete and inefficient fat oxidation? Well, there could be an imbalance in the Krebs cycle I mentioned to you earlier, where its capacity or its activity is lower than what might be optimal to allow the fat to be fully burned. There could be increased delivery of the fatty acids, either within the tissues, being liberated within the tissue, or being delivered through lipolysis or from the fat tissue during an activity or fasting or these sorts of conditions. The oxidation of the fat or the presence of the fat might be mismatched to the ATP production. So if there's an issue with the mitochondrial ATP production, you might imagine that the fuel delivery can't match the fuel production, and this would cause increased sort of smog. Also remember that under conditions in which oxygen is lower, for example, in a condition called ischemia of certain tissues such as heart, when blood flow is reduced, the concentration of oxygen is obviously reduced within that tissue. That's called PO2. PO2, or partial pressure of oxygen, is reduced. This also, by the way, happens naturally in our muscles when we exercise. So any of us who would get on a treadmill right now and would ramp up to 20, 40, 60% of our maximum energy or maximal oxygen consumption and exercise, actually, if you measured the partial pressure of oxygen within our tissues, it actually drops pretty remarkably. You know, I sort of think it's one or more of the above, and I think it's context dependent why there would be these inefficiencies present in any one particular moment in time. So here's that TCA cycle again. You may remember this process here. The fat is burned, and when it's fully combusted, it goes through the Krebs cycle here. But to make that happen, the Krebs cycle actually needs something called anaplerosis. And this is to keep it humming because it's always losing carbon. And when you're losing carbon out of the furnace, you can't burn the fuel trying to come into the furnace. So anaplerosis is a way to get more carbon back into the cycle. And this can come from a variety of places, from pyruvate, a couple of amino acids, and then you heard earlier about some of these odd chain fatty acids or triheptanoin. That's a C7, so an odd chain fatty acid. These sorts of things can be broken down through this pathway to propionyl-CoA and ultimately make their way in as succinyl-CoA. So I suppose that the triheptanoin in part must be working, I suppose, through anaplerosis and delivering carbon to the Krebs cycle to help overall metabolism. The importance of this can be shown here. So these are concentrations of different acylcarnitines, long-chain acylcarnitines here, here, and here. These are muscle mitochondria isolated from rats. And what you see is when you're missing malate from the mix, malate is right here. When you're missing this, you get a large accumulation of acylcarnitines in the media around these mitochondria. What that means is the fat has no place to go. The long-chain fatty acids have no place to go, and there's a whole increase in acylcarnitines more generally when there's not enough of an anaplerotic environment. There's a different way, a different thing that could be influencing the Krebs cycle. It's called cataplerosis. It's almost like the opposite of anaplerosis. It's actually the loss of carbon from the TCA cycle. So it's actually not particularly well studied, but it could be another contributor to inefficient fat metabolism and buildup of smog, at least in my mind. So in this case, again, here's the Krebs cycle. Here's fatty acid oxidation delivering some acetyl-CoA to the mix. It turns out that several of these metabolites in the Krebs cycle are actually exported in the process of fatty acid oxidation, and including inefficient fatty acid oxidation. So while the reasons for this are not entirely clear, at least not to me, when you're combusting fat, inefficient or efficient, you actually export the very things you need in terms of some carbons in the Krebs cycle to maintain an efficient metabolism. This is an example in rat hearts, a study some years ago, and I want you to go right to left with me here. This is citrate, which you may remember is right here. This is the concentration basically released from the heart under conditions of high fat or lower fat concentration in the perfusion buffer. So when you go to higher amounts of fat, you actually export increased amounts of citrate from that tissue. Here's a study we did a number of years ago in isolated mitochondria from mice. And when we increase the amount of fatty acid concentration from left to right, we increased fatty acid oxidation. I don't show that here, but we did. And what was interesting was citrate was exported. This is the concentration in the buffer around the mitochondria. Almost immediately you saw an export of citrate with fatty acid oxidation. When we added an inhibitor of fatty acid oxidation here, we completely blocked this export. So this is very interesting. I actually can't tell you what the ramifications are for this in vivo, but we've speculated that an imbalance between anaplerosis and cataplerosis can occur under certain conditions, and certainly this would back up the system and cause an increased amount of smog to accumulate. So, another question. Regardless of the origins of the mismatch between fatty acid oxidation and full combustion of the fuel in the Krebs cycle, are there specific metabolites that accumulate and trigger a pro-inflammatory or cell stress event? We kind of had a clue in our early days of thinking about this from work that had been done with saturated fatty acids. So these are just things like palmitate or other fats of that nature. And on the cell membrane, there are a series of receptors called Toll-like receptors, or TLR4, TLR2, and others. These actually are constantly sensing the environment, looking for things like bacterial components and other sort of pathogens to allow us to, in our immune system, to kind of activate a whole cascade of events to increase inflammation. And the reason this is there is to be helpful. In other words, you want your body to respond, the immune system, with inflammation in response to pathogens. But what's interesting is there's a whole body of work to suggest that certain fatty acids actually can also activate this pathway in a system called sterile inflammation. So we naturally began to think, well, wouldn't it be interesting if acylcarnitines, which is part of the smog that occurs with inefficient fat oxidation, might these be activating the inflammatory process? And our early work, and I think our more recent work, suggests absolutely. So this is a reporter called NF-kappa B. This is just a downstream factor in that cascade I showed you a second ago. And the question here is, if you add these different components including this fatty acid here and this fatty acid here, C12, C14, but now they're carnitine esters shown here. Can you activate inflammation as judged by this reporter? And the answer is absolutely you can. By the way, this is just a positive control endotoxin from bacteria that's often used in these sorts of studies to trigger inflammation. So the point being, acylcarnitines from this very early study absolutely could potentially trigger inflammation. We moved on from that and looked at other inflammatory events in macrophages, which are kind of one of the attack cells in our body. And we found that with increasing chain length of acylcarnitines, we could trigger this marker called COX-2, which is an inflammation marker. And this is the protein level. So you see here, you really start to see things at C14, C16, and C18. But we've also seen activation with C12 and C10 in terms of this marker. These are just different concentrations of C14 carnitine. We typically see changes in inflammation at as low as between sort of 2.5 and 5 micromolar of this particular acylcarnitine. These are different cytokines which are put out by immune cells to help battle pathogens, typically to alert other immune cells, hey, we need some help in here. This is TNF-alpha, MIP2, and MCP1 as examples. This is, again, C14 carnitine shown here in different concentrations. And again, by certainly 5 micromolar, we begin to see an activation of inflammation. And these high bars here are just positive controls. These are bacterial components that we use in the system just to make sure, we're on track and the system is working to activate inflammation. So back to this schematic here, what is the mechanism of acylcarnitines activating inflammation? And we first ask, well, logically, maybe it's one of these receptors, one of these toll-like receptors that we know fatty acids can activate. Maybe acylcarnitines activate those as well. So we did a panel shown here. So this is a screen for different toll-like receptors. Here's all the different names of the different toll-like receptors. This is C14 carnitine we used. And this is the fold difference relative to just basal. And you can see this is about no change. One fold difference is the same as no change. Contrast that with positive controls from bacteria and other different sources that show a very strong effect or positive effect on activating these receptors. So that had us scratch our head because we certainly have no evidence that it's these receptors that are involved with acylcarnitine activation of inflammation. Okay, well, what else might it be? So we looked a little further downstream at this molecule called MyD88. This is a cofactor for many of these different pathways. In other words, it's kind of a policeman or a lieutenant for these different commanders to activate this pathway in many cases. What role might it play? Well, what we did was we knocked it down through genetic means in macrophages. So we basically got rid of the protein. This is MyD88 protein here. It went away compared to the control cells shown here. And what was interesting was this marker of inflammation in response to C14 carnitine, which was activated by C14 carnitine, that response completely went away as well. And this indicates that without MyD88, you lose the effect of the acylcarnitine to activate inflammation. We recently did a gene expression study where we treated cells, macrophages, with acylcarnitine, in this case C14 carnitine at 5 micromolar. Here's another control here which activates a TLR4, TLR2. Here's another factor here, positive control that activates TLR2. And here's C14 carnitine treating macrophages. And these are simply gene expression differences looking for effects of the C14 carnitine. And certainly there are a number of genes which are upregulated or downregulated in response to acylcarnitine in these immune cells. Some of them are shared with these toll-like receptor activators. But in the case of MyD88 knockdown, where we get rid of it, no response whatsoever at the gene expression level for acylcarnitine. These were very complex patterns, which I want to definitely ignore for the time being. These are preliminary data. But I think it makes the point that acylcarnitines can activate inflammation, and this is an MyD88 dependent matter. A second pathway that's very, very interesting, and I think I'll hopefully prove this to you in a minute, is this system called caspase 3. And if you'll be patient with me, I'll show you what the heck caspase 3 is in just a second. But I can assure you that it's a cell stress and even a cell death marker. So when it's activated in a cell by a variety of different stimuli, it becomes cleaved. And you can pick that up in this sort of protein assay here. This is the number of minutes of time. And this one would be in muscle cells. And this in this case, it's C16 carnitine. So it is a little different. It's Palmitoylcarnitine, long-chain acylcarnitine. And you can see by even 10 minutes of treatment with C16 carnitine, you can activate caspase 3. These are positive controls over here. Again, things we know activate caspase 3, and it just shows that the system is working. Earlier, someone else, Mutumba et al., had taken some purified caspase 3 protein and had applied palmitoylcarnitine to it and found that you could activate the cleavage just by putting them together sort of in a test tube. So this is really, really interesting, and to me indicates, I think, very clearly that acylcarnitines have the potential to activate cell stress cascades and perhaps even activate cell death in the right context. It's just a hypothesis. So here's caspase 3. Here's just a review article from a different group showing that after a whole series of stimuli, including sort of damage to the cell or other pro-inflammatory signals shown here, you could activate caspase 3, and it can go into activating a whole series of events which can either kill the cell or certainly can cause stress in the cell. Here's another example, a more recent paper showing that active caspase 3 outside the mitochondrion can make its way into the mitochondrion and actually have a negative effect on bioenergetics and ATP production. So I think you're getting the message that perhaps some of these pathways could possibly be activated under the right conditions, and maybe this is helping to trigger some of the issues with rhabdomyolysis and other things of this nature. Okay, take-home messages, which I probably already just said. Long and medium chain acylcarnitines do have the potential to activate inflammation in macrophages. Interestingly, these do not require those toll-like receptors, and they are impacted by the presence or absence of this very important protein MyD88. Some of our more recent data, which I'm not ready to show yet, would suggest that in the absence of MyD88, there is a receptor that might be binding acylcarnitines and possibly triggering this whole cascade of events I mentioned to you earlier. That's highly speculative. It's something we're working on now. Activities in the muscle and the heart are less clear, but potentially caspase 3 may be playing a role here to activate cell stress and mitochondrial dysfunction under the right conditions of acylcarnitine accumulation. We also know that acylcarnitines activate intracellular calcium mobilization, and this is probably involved in acylcarnitine signaling. We published some of that work. So let's revisit our question. Do specific metabolites that accumulate in FAOD under certain conditions trigger pro-inflammatory and cell stress events? I would say from the data I've shown you so far, and where my brain is at least, acylcarnitines are plausible candidates, definitely. So I want to take you on another brief journey about heart ischemia. And the reason I'm going to take you there is because I think this is a great example of how acylcarnitines, I think this cements the idea, I hope, that acylcarnitines absolutely could be part of the problem in these episodic conditions that lead to rhabdomyolysis and other myopathies. Here's an example where this team by Lipids et al., and all the work I'm going to show you is from that group, blocked a carnitine transporter, shown here, with different pharmacologic agents. They named them 4, 16, 39, 43. I don't know why. But what they did was they gave the system, these were isolated rat hearts, they perfused them, or pardon me, they treated the animals with these inhibitors first. Then they took the hearts and they looked at the amount of carnitine and acylcarnitine in the heart. This particular compound dramatically lowered the amount of carnitine in that tissue. And what was interesting was they were able to lower the infarct or the cell death size in the heart by 40% when they blocked blood flow for a period of time. Blocking blood flow is called ischemia, and in this case it was done experimentally. The point being it can kill cells, as you might imagine, if it's done long enough. But what was interesting was when there was very little carnitine on board, it turns out you could protect the heart compared to other conditions. This is another treatment with kind of a drug that has lots of different effects, so it's a little bit of a dirty interpretation. But if you treat animals with this drug called mildronate, you can actually lower, again, the concentration of carnitine in the heart. You can then do the same study, do an ischemia model where you block the blood flow to the heart for a period of time, and then you looked at the infarct size or the cell death, tissue death. And again, a reduction. So these are all associations which would suggest that if you limit the amount of carnitine around, and they've shown this lowers the acylcarnitine concentration dramatically as well, you can actually have a protective effect on cell death under conditions in which the system has been engaged and is under tremendous stress. Here's another experiment shown here. If you look in an ischemia model, this is the concentration of long-chain acylcarnitines and long-chain acyl-CoA in the heart, in the at-risk area, the area that's been affected by ischemia, and the area that's not been affected by ischemia. And what you see is a tremendous increase in these metabolites in the stressed tissue. When you look at mitochondrial protein shown here, you get the same story. And what's particularly interesting, they then looked at the mitochondrial pieces, if you will, here, and looked at the concentration or the amount of accumulation of this radio-labeled acylcarnitine. The outer mitochondrial membrane hardly had any at all. Same thing for inside the mitochondria. But in this inner leaf, if you remember the schematic I showed you earlier of CPT1 and CPT2, the electron transport chain where all that ATP is made, there's tremendous increase of accumulation of acylcarnitine in that specific spot. Now again, if you take a heart, in this case, and you infuse it with palmitoylcarnitine or acylcarnitine, you can actually get an accumulation of acylcarnitine, long-chain acylcarnitine in the mitochondrion. I don't show that here from their study. But what happens when you do that, you also get an increase in cell and tissue damage. Here's something they looked at cardiac work, and while they were infusing the palmitoylcarnitine in these rat hearts, they found that the ability to do work was lower in the presence of palmitoylcarnitine than when no palmitoylcarnitine was infused at all. Again, to me, all of these are suggesting the potential that under the right condition, or you could say the wrong condition, the presence of long-chain acylcarnitines and perhaps chain-shortened acylcarnitines in these tissues could exacerbate or trigger cell damage, tissue damage, and a lower function bioenergetically. So why might this be the case? We have proposed in at least a couple of papers that it may be because these long-chain acylcarnitines are really, in a way, detergents. Their structure has this plus charge and this minus charge. This is palmitoylcarnitine, which looks very similar to something called phosphatidylcholine and other similar molecules in the body. These actually act sort of like detergents because this part of the molecule shown here without any pluses or minuses is the true lipid portion or the fat portion, which you could consider sort of greasy. This is the charge portion, and these embed themselves into membranes very, very readily. And this could be in a pH-dependent manner. So we've proposed, and others have sort of alluded to this as well, is that when these acylcarnitines accumulate too much, they can figure out a way to get into the mitochondrial membrane and the cell membrane and other membranes and disrupt the function of the cell. Here's another example, again, from Lipinch's group. This is oxidative phosphorylation. So earlier, you remember we were talking about taking fuels, burning fuels, and making ATP. These are isolated rat mitochondria, and they're under conditions in which they're using oxygen, they're making ATP. But look what happens when you start to add more and more palmitoylcarnitine. All of those processes, and you can ignore these for just a second, all of these processes are diminished in the presence of increasing amounts of palmitoylcarnitine. So it's possible that as these accumulate under certain conditions, that it could be impairing the bioenergetic health of the cell. What's interesting here is what they did, they threw in some binding proteins here. This is called acyl-CoA binding protein. This is called fatty acid binding protein. If you could bind up the acylcarnitines using these proteins, could you rescue these mitochondria so that they would continue to burn oxygen and burn fuel and make ATP? The answer was no. But what they did find was that if they took a fraction of the cell that contains other proteins, shown here in this square, compared to this one here, you see they are able to maintain their bioenergetics much better, leading to the postulate that these acylcarnitines can be bound by certain proteins in the cell, and perhaps these proteins are protecting the cell or protecting the tissue from too much free acylcarnitine. And I'm going to show you I've shown this one before. If it works, we'll soon find out. This is a video I'll show you of acylcarnitines. This is palmitoylcarnitine. It turns out these bind very, very well to that protein you heard earlier called myoglobin, but only in the oxygenated form. I don't think my movie's going to work, but I can assure you that, so this is a picture of the myoglobin here, a 3D format, and this is a palmitoylcarnitine. And what's interesting is this palmitoylcarnitine winds its way into the myoglobin and sticks there. And we've shown this published this in several papers, that in the oxygenated form, shown here by these two red dots, oxygenated myoglobin can bind very avidly acylcarnitines. And that may come into play in a minute as I tell you a little bit more of the story. Note that it cannot bind to deoxymyoglobin. And so under conditions of ischemia, or when the PO2 or the partial pressure of oxygen drops in a tissue, myoglobin becomes deoxygenated. It loses its oxygen. And what's interesting is there would be no way for the acylcarnitine to bind in that condition. We've sort of thought, well, maybe it's a situation in which the acylcarnitines can become released, and that can maybe be a bad thing. So here's a working model. Here's what I'm thinking. Under conditions of heart ischemia, hypoxia or low oxygen lowers fatty acid oxidation because there's not enough oxygen to burn the fuel, but it does not inhibit carnitine palmitoyltransferase 1. So carnitine palmitoyltransferase 1 can continue to make long-chain acylcarnitines. No problem at all. As long as there's carnitine around and fatty acids around, CPT1 will make acylcarnitine. This leads to an accumulation of these long-chain acylcarnitines that I showed you earlier in the heart model. Theoretically, this could inhibit mitochondrial function through different membrane activities, electron transport chain. There's certain ion channels which are disrupted, and it can activate caspase 3, that cell death sort of inflammation and cell stress pathway I mentioned to you earlier. And this could, in theory, exacerbate infarct in the heart or tissue death. And under conditions of low oxygen, oxymyoglobin is converted to deoxymyoglobin. It cannot bind acylcarnitines under that condition to serve as a reservoir to kind of hold them back. And what's interesting, I won't have time to talk about it here, deoxymyoglobin actually also produces something called nitric oxide. So in parallel, nitric oxide, it could be produced, and this inhibits the electron transport chain as well, again, exacerbating the smog further and further as you go along. What about FAODs? Is there any parallel path here? And I think there may be, and this is what I'd like to propose today. We talked a little bit earlier that in certain FAOD conditions, CPT1 continues to work just fine, despite the lesion in the enzymes lower in the pathway. Perhaps under very specific conditions, infection, catabolic states, excess exercise, undernutrition, inflammation, lots of different things that remain to be worked out, perhaps this causes an increase in the smog. And perhaps this causes a backup in the system enough to accumulate long-chain acylcarnitines and some medium-chain acylcarnitines, which in turn, just like in cardiac ischemia, can cause a dysfunction of mitochondria, activation of caspase 3, and then perhaps even inflammation cascades in immune cells and other cells. If this is all true, and I have no idea if it is, this would tend to exacerbate myopathy or trigger myopathy. It could kind of explain the low energy, the feeling of low energy that folks have when they have these episodes. And it could even cause cell death, which would lead to rhabdomyolysis and other symptoms of this nature. And again, because oxymyoglobin is an acylcarnitine binding protein, under these conditions in which there is perhaps some change in oxygenation or other factors, maybe myoglobin is playing a role here as well to exacerbate the problem. I don't know. This complicated slide we published, so if you're interested in looking it up or following up with an email, let me know. These little things here we're calling long-chain acylcarnitines. This is just a cell, let's call it a heart. And you can see with excess accumulation, what we proposed was that you would get an increase in cell permeability and cell death, which could lead to the CK increasing that we heard about earlier. Definitely ion channels that deal with potassium, calcium, sodium would be disrupted, absolutely. There would be cell stress and cell inflammation cascades. And then, of course, as I mentioned to you, the very likely possibility that even mitochondrial function is disrupted. So let's revisit our question as we head to the finish line here. The question was, do specific metabolites that accumulate in FAOD, at least under very specific conditions, trigger pro-inflammation cell stress events? I'm changing officially today. The acylcarnitines are likely candidates. Forever it's been plausible. I think they're very likely candidates to contribute to some of these pathologies, again, under very select conditions. So some very, very speculative future directions. Some of these might get me in trouble, but we'll see. Question: does lipotoxicity only manifest in the presence of other triggers? This was alluded to earlier by Dr. Corson, where we really don't know what causes these episodes to come on. I would argue that everything I've just shown you above could take place, but it has to be under just the right conditions. I'm actually not sure what those would be exactly, but I think some of those trigger points like inflammation and perhaps oxygenation, too much exercise, hypoxia could play a role. So what are the mechanisms that link this molecule MyD88 to acylcarnitine-induced inflammation? I don't know. But if we could figure that out, it might be a focus of future FAOD treatments. If we could understand how to manipulate that system better, maybe, maybe not, it could lead to new treatments. This caspase 3 effect, this cell stress effect, is very intriguing, very convincing actually. The question would be, could a caspase 3 inhibitor be explored as an FAOD treatment? And I'm not necessarily saying as a chronic treatment, but under these episodes or where these episodes are coming on, might this be a helpful adjunct to other treatments? If this hypothesis about myoglobin as a binding protein is true, it's really interesting to think that its protective role as a binder goes away when there's ischemia or hypoxia, or perhaps when there's been too much exercise and the myoglobin becomes deoxygenated. This is purely speculative, but very, very intriguing to consider. Here's a very controversial question. Do the findings in heart vis-a-vis lowering tissue carnitine have any relevance to FAOD? Typically, of course, carnitine is one of the drugs that's used to help people stay healthy. The question really is more, as these episodes come on, is there any value to considering limiting or lowering the acylcarnitine accumulation in tissues through some maneuver that lowers tissue carnitine? I'm not sure about that one, but I thought I'd raise it as a question. And then finally, I've mentioned to you acylcarnitines because it's something we study. But I think we really need to look beyond acylcarnitines and ask questions. Are there other lipids that contribute to this sort of lipotoxicity that leads to myopathy in heart tissues and cardiomyopathy? There is some evidence, for example, of long-chain acyl-CoA causing some issues, ceramides, sphingomyelin. There's a lot of other things that go on with lipids beyond acylcarnitines. And so I definitely have an open mind, and it may be a combination of many things. And the reason I say this, and it's the last data slide, is this one here. This was a collaboration with Dr. Gillingham, Dr. Vockley. If you look at blood in patients who are harboring these fatty acid oxidation disorders, and you look at their blood and you compare it to control blood, what you see is a whole variety of lipids. These are all funny names for very complex lipids. And I believe you're going to be hearing about cardiolipin tomorrow. But my point is, these are complex lipids in the blood, and you can see some of them are elevated in folks with these disorders, and some are reduced. But certainly, there's a huge difference between persons who are healthy and do not have an FAOD versus folks who do have an FAOD, even in their plasma or their blood lipid profile. So what's going on in the tissues? We don't know. But I think this tells us we maybe need to look beyond acylcarnitines as we consider lipotoxicity as part of the picture and the landscape of FAOD. So science is a team effort. Before I close, I want to thank a lot of the work that I showed you was done by these three individuals, Kikumi, Michael, and Sri. Previously, two of them at UC Davis, and then we moved to Arkansas. And then some folks at UC Davis, Trina and Colin. And of course, my collaborators, Jerry and Melanie, who are just wonderful, wonderful collaborators in this area. So with that, thank my funders, and thank you all very much for allowing me this opportunity to speak today. Thank you.
Thank you so much, Dr. Adams. That was all great information. It's great how everything's just rolling. One presentation's rolling into the other, and it's all connected, which is good. So we've got a few questions before we sign off here. First question is, does this inoptimal oxidation of fat lead to higher HD cholesterol?
Let me make sure I've got I had to adjust my speaker there.
Okay.
Was the question related to, does this impact cholesterol?
HD cholesterol, higher HD cholesterol.
Okay, gotcha. Yeah, I would say I don't know. That's going to be my pat answer.
We'll take that. We'll take that.
I don't recall in speaking with Melanie, Dr. Gillingham, for example, that the patients we were looking at for those studies of all those lipids I showed you, I don't recall that there was anything particularly different in HDL. So those were remember what I showed you, those were profiles. Think of them as landscape or a painting. Those are a profile of the lipids that are there. It doesn't automatically mean you have either higher amounts of say HDL or not. So, but I don't remember any difference between those two groups.
Okay, great, thank you. Next question is, if acylcarnitines make a sterile inflammation, do NSAIDs such as ibuprofen help better with metabolic crisis than, for example, acetaminophen, even though NSAIDs are potentially harmful to the kidneys?
Okay, I do wish Dr. Vockley were here, but my short answer is we don't know, right? So what I've shown you is the real data, but these are speculative in terms of their application to the episodes that get kids, into the ER. So one thing that Dr. Vockley and I have talked about for many years is to better understand when there's a crisis that leads to rhabdomyolysis or, as things are leading up to that episode that ends up with a hospitalization, what are the inflammatory profiles in those kids? all we have at this point is sort of anecdotal information. We don't have any study that's very, very specifically looked at whether or not there's a cytokine or an inflammation surge in certain kids with those incidents, yes or no. So I think we need to know that first before we start thinking about, well, maybe we need to start limiting inflammation because that'll help with the episode.
Right.
I don't think we know yet.
Okay, great answer. Okay, what makes inflammation a positive thing in your body versus the negative implications we think about?
Can you repeat that, Kyra, please?
What makes inflammation a positive thing in your body versus the negative implications that we normally think about?
Yeah, okay, great. So obviously, if you don't have an immune system and you don't have an inflammatory response, you're going to be, there was an old movie from the 70s, The Boy in the Plastic Bubble. You're really going to have trouble fending off infections, diseases, and you really have to isolate yourself completely. So this tells you that there's this natural critical role for the immune system just to take care of pathogens that we see every day. In addition, the immune system has a critical role in cancer surveillance. So the immune system is constantly surveilling the body for things that are not like me. And that could be cancer cells. And in fact, the immune system can often, I've heard in different talks, that takes care of cancers every day that we never know about. We live a long, healthy life because of it. And when that's compromised, however, you are more susceptible to certain cancers. So there's a lot of good, right? Oh, and when you heal, if you cut your hand or something happens and you need to heal, the first cells that tend to be in there very often are macrophages. Those cells I was telling you about earlier that show a pro-inflammatory effect against the lipids. Those are natural, important remodelers. They do a great job of remodeling that tissue. So those are all good things. Where things can go south is a perfect example is COVID-19. I hate to bring up COVID-19, but the cytokine storm, they call it, is where things get out of control. And this can cause then cellular damage. It can cause a whole cascade of events in different tissues as those cytokines are recognized by receptors and cause cascades of cell stress events throughout the body, including the heart, by the way. So that's when it's not good. And so I guess what I'm proposing here would be, well, maybe under very certain conditions, some of these pathways get engaged too much, whether it's the caspase pathway, whether it's inflammation, whether it's both. And maybe that helps exacerbate some of the pathologies or some of the symptomology.
Good, yeah. So inflammation is not always a bad thing.
No, inflammation can be very good.
Okay, next, and this probably will be our last question, is how do high acylcarnitines affect the gut and gut bacteria? And could this be why some of our kids have issues with their gut?
Oh, man, that's a loaded question. Okay. Kyra, this may be something you and I are talking about next week, too.
Okay.
But anyway, as I mentioned, no idea. Okay, no idea. So host metabolites do get into the gut, okay? So we know this. It's kind of underappreciated, actually. So things coursing through our bloodstream absolutely can make their way into the gut, literally into the gut lumen. I have no clue if anybody's ever looked at whether or not, acylcarnitines, make their way into the gut or the gut wall to in turn somehow have a discussion with the microbes. That's a very innovative question. I've spoken with Drs. Gillingham and Vauckley a little bit about perhaps a related but a separate question on the gut microbiome's role in the health of people affected with this disorder, this disease, and the different diets people are on and that sort of thing. Totally unexplored. It's not exactly answering the question, but that area alone is, I think, really, really important. What's happening in the GI tract of these kids and young people who have the disorder and are perhaps on a completely different kind of diet. Does this change the microbial ecology? I think it will. And then does that in turn cause a change in host health? So this is something we do study in my lab, and we're pretty excited about the possibility of studying that question in collaboration with others. It's just going to be a matter of funding and a matter of having a look.
Yeah. Well, clearly we have some budding scientists in our audience who are asking these great questions. And Dr. Adams, thank you so much. I mean, there is a lot of conversation to continue to have. We look forward to continuing to hear more about your studies and all that you're doing. And we'll stay in touch, and we look forward to hearing more from you. Thank you so much for being with us today.
Thank you all for the opportunity.
Thank you.